So today, children, we are going to start with the new topic, and the topic is chemiosmotic hypothesis. Till yesterday, we did the cyclic and non-cyclic photophosphorylation. We did cyclic and non-cyclic photophosphorylation. In this, we learned that in cyclic, only ATP is synthesized, whereas in non-cyclic, ATP and NADPH2 both are synthesized. Here only one, here these two high energy compounds are synthesized. Now, what we are going to do today is chemiosmotic hypothesis or process of chemiosmosis. Now, how this is linked with chemiosmosis? Chemiosmosis basically is the process of chemiosmotic hypothesis. Basically, it tells about the process of synthesis of these high energy compounds. Both. Process of synthesis of ATP and NADPH. This process of synthesis is explained by chemiosmotic hypothesis. Now, this process of synthesis of ATP and NADPH2 or process of synthesis of high energy compounds that is ATP and NADPH. This takes place in two organelles. This takes place in two organelles. This process is seen in two organelles. The first organelle in which this process of synthesis of ATP and NADP is seen is mitochondria. And the second organelle where this synthesis of high energy compound is seen is chloroplast. So these are the two organelles where the synthesis of high energy compounds is seen. Now, in mitochondria, this takes place in intermembrane space. The site of synthesis is, in case of mitochondria, it is intermembrane space. Whereas the site of synthesis of high energy compound in case of chloroplast, it is thylakoid membrane. If this is mitochondria, we have crystals in mitochondria like this. There are crystals in mitochondria. Where does this synthesis of high energy compound take place? It is here, this area, where the synthesis of high energy compounds takes place. And in case of chloroplast, if we see chloroplast is like this, we have stakes of thylakoids arranged one over other. Connected by lamellae, stroma lamellae. And this is the thylakoid. This is thylakoid where the process of synthesis of high energy compounds takes place. So in both organelles, The high energy compounds ATP and NADPH is synthesized, and this is the location where these are synthesized. Now children, we will start with chemiosmotic hypothesis, or the process of chemiosmosis, which is seen in the synthesis of high energy compound in case of chloroplast. So for this we will have to draw a thylakoid. Let's draw a thylakoid. This is a thylakoid, the inner membrane and the outer membrane. We have to draw our inner membrane. This is a thylakoid which has the inner membrane and outer membrane. And as we know that in thylakoid, photolysis of water also takes place and formation of NADPH also takes place. The enzyme required for the formation of NADPH is NADPH reductase. So photolysis of water takes place here inside, you know, on the inner membrane. Let me draw it with another color. Suppose this is the side, this is the complex for photolysis of water. And this is the place where NADPH reductase enzyme is present. NADPH reductase is present on the outer membrane, and photolysis of water, the complex which helps in photolysis of water, is present on the inner membrane of thylakoid. So this is a structure of thylakoid, which shows an outer membrane, and inner membrane, now we have to study the synthesis of ATP and NADPH. So it takes place in non-cyclic photophosphorylation. So we will, in non-cyclic photophosphorylation, two photosystems are there, photosystem 2 and photosystem 1. Both the photosystem are operative in case of non-cyclic photophosphorylation. So that we are going to make. In the NCERT book, this picture is given very clearly. So here let's make the photosystem 1. This is photosystem 1, or we can say that this is PS1. PS1, photosystem 1. Now we have photosystem, in case of non-cyclic, first it is photosystem 2. Then we have photosystem 1. Let's make photosystem 1 here. PS1 is located here. So when the electron is excited from PS2, it jumps out and is accepted by primary electron acceptor, and then it moves downhill when the energy is released, and it is received by various electrons acceptors in the electron transport system. When we did non-cyclic, we studied there were three complexes of electron transport system. First was plastoquinone, then cytochrome, and then plastocyanin. So if you remember, those three complexes were there when we did non-cyclic, we did that. So the most important out of that is cytochrome. So we are going to make cytochrome here. So this is the middle complex of electron transport system. which is cytochrome. So we have made cytochrome here. This is cytochrome, and specifically two types of cytochrome are there, B and F. Okay, so this is now cytochrome, and this forms the non-cyclic system, or the machinery involved in non-cyclic photophosphorylation has been shown here. Now beta, basically the synthesis of ATP and NADPH is based on one principle, that is proton gradient. It is based on this principle of proton gradient. Whether it's mitochondria or chloroplast, always the synthesis of ATP and NADPH is because of this proton gradient. Now, what happens in this case? There should be a proton gradient developed. That means the number of protons inside this lumen, number of protons here, should be more as compared to protons which are present in the stroma. That means protons inside the lumen should be more as compared to the protons which are present outside the thylakoid in the stroma. Then this will develop a proton gradient, and when this proton gradient will be broken, energy will be released, and that energy will be utilized for the synthesis of ATP. Basically, what we are studying right now is the synthesis of ATP. Now, how this proton gradient is developed, that means how the number of protons are made more inside the thylakoid as compared to in the stroma. This inside the thylakoid, this space which is inside the thylakoid is known as lumen. Any cavity is known as lumen. The other day also I was telling you that it is lumen. The inside cavity is lumen. Now what happens when we did the process of non-cyclic photophosphorylation, we studied that PS2, in the PS2, when in the reaction center, the sunlight reaches, chlorophyll A absorbs it, and then the other accessory pigments also absorb light, and then that is transferred to the chlorophyll A, and when that light is transferred to the chlorophyll A, then it a pair of electron in chlorophyll A gets excited, and it comes out of the shell where it is accepted by a primary electron acceptor. So this is what we studied. PS2 was there. I'll help you in understanding this. PS2 was there. An electron jumps up, primary electron acceptor, and then it goes downhill. First electron acceptor, second electron acceptor, third electron acceptor, and then it moves to PS1. Again jumps up, primary electron acceptor, and then moves downhill where NADPH is formed. Whereas here it is formed at the cytochrome level. ATP is formed here. This is what we studied in non-cyclic photophosphorylation. The same thing is happening here. The same thing is going to happen here in this. Now, how the concentration of protons is increased inside the lumen, or how the H plus proton, H plus, that means protons, their concentration is increased in the lumen of the thylakoid. This is what we are going to study now. This process was just a reminder to you so that you the process you remember it properly, what happens in non-cyclic photophosphorylation. Now here, the electron jumps. The electron jumps when it gets excited due to accumulation of energy. Okay, it jumps and is received by primary electron acceptor. This is what happens, primary electron acceptor, and then it starts moving downhill. So when it moves downhill, it is accepted by various electron acceptors. It is accepted by various electron acceptors. The most important one is cytochrome. Now, what happens here at cytochrome? See, basically we say that cytochrome is also an electron acceptor, but it is not an electron acceptor. This cytochrome is an electron transporter. The correct word which we can use for this is transporter. Electron transporter. So the pair of electron which moves downhill reaches cytochrome. Now this cytochrome, actually when it reaches cytochrome, it wants to transport it to PS1. Now how will it transport? It doesn't transport the electron through an electron acceptor. Rather, that electron which has to be transported from here to PS1 is transported by a proton. A proton is used to transport the excited electron from cytochrome to PS1. And from where this proton comes, which carries that electron, excited electron, from cytochrome to PS1, from where that electron comes? That electron comes from the stroma. This is stroma. Outside thylakoid is stroma. This is thylakoid. This is thylakoid, as you know. Thylakoid. And this is outside is stroma. So what happens that it is accepted, the electron which has now started moving downhill, is at the cytochrome is accepted by a proton acceptor, a hydrogen acceptor or a proton acceptor. Now from where this proton comes? This proton comes from the stroma. That means the cytochrome will take one hydrogen from here. This will accept an electron, and then this acceptor, this has taken hydrogen, proton, which accepts this electron, and then this electron is transferred to this, the pair of electron is transferred to PS1 system, and this proton goes in the lumen of the thylakoid. Proton goes in the lumen of the thylakoid, whereas the pair of electron is transported to PS1 system. Okay, that means from the stroma, the protons are being utilized by the cytochrome to transport excited electrons. That means outside in the stroma, the concentration of proton is going down and the number of protons are increasing in the lumen of the thylakoid. In the lumen of the thylakoid, the concentration of proton is increasing by this method. Is it clear to you, children? Is this point clear to you, or shall I repeat? Yes, beta. Is this point clear to you? The first point, why the concentration of protons inside the lumen of the thylakoid increase. Okay, Yashika is saying it's clear. Yes, okay. Yeah, okay, clear. Ananya is also saying clear. Yes. Deepika, fine, it's clear. Okay, so this is the first way by which the electrons, sorry, not the electrons, protons, their number increases inside the lumen of the thylakoid. This is the first method. That means while transporting electron through the cytochrome, it is a hydrogen acceptor which transports it here. After transporting the excited electron to PS1, that hydrogen acceptor which comes from the stroma doesn't go back to stroma, rather it comes to the lumen of the thylakoid, thus increasing the protons in the lumen of the thylakoid. This is the first way for maintaining proton gradient. That means increasing the number of protons inside the lumen. Now the second way. What is that second process by which the number of protons increase inside the lumen? The second is photolysis of water. I told you this is a complex. This is a complex, this black color which I have made. I'm erasing this thylakoid. I'll write here thylakoid, but I think I should write it inside so that you understand that this is thylakoid and that is stroma. So this is a complex which helps in photo. This is a complex. This black color one is a complex which helps in photolysis of water. Photolysis of water. Now what happens because of photolysis of water? This is a complex which helps in photolysis of water. That means H2O getting broken down to form electrons, to form protons, and to form oxygen. This is what is happening as a result of photolysis of water. Now the oxygen goes into the air. From here the oxygen is released into the air. The electrons keep on joining these electrons. Here these electrons, they join the PS2 system in restoring the lost electrons. These, see, these, the oxygen goes into the air. The electrons from here, they join PS2 for restoring the electrons which are lost as a result of the non-cyclic photophosphorylation. The third, which are left here, are protons. Where will these protons go? These protons stay inside only. These protons, they stay inside in the lumen. That means this is the second way by which the concentration of protons increase inside the lumen of thylakoid. This is the second method because the protons are not going anywhere. Electrons, they join PS2, oxygen is released into the air. The protons, they remain in the lumen of the thylakoid, thus increasing the concentration of protons in the lumen of thylakoid. So this is the second way by which the protons increase inside the lumen of the thylakoid. Now the third way by which the proton inside the lumen of the thylakoid increase and the protons in the stroma decrease. What is the third way? Third way is this. See, the first way is this. This is the first way which I have told for increasing the number of this protons in the lumen. This was the second way. Now the third way is this, where I am writing third. This is the first way, this is the second way, and the third way. This is the third way by which the protons increase inside the lumen and decrease in the stroma. What is this black structure which I made? This is the position of NADP reductase enzyme. This is the location of NADP reductase enzyme. This NADP reductase is an enzyme which is required to convert NADP into NADPH2. This is the enzyme which is required to convert NADP into NADPH2. So, see, when NADP gets converted into NADPH2, what is taking place? Addition of hydrogen. When the NADP is getting converted into NADPH2, reduction is taking place. That means hydrogen is being added. Now, from where this hydrogen will come? From where this hydrogen will come? It will come from the stroma, from this outside stroma region. This stroma. This hydrogen which is required for the reduction of NADP to NADPH2, it comes from the stroma. Now, when it comes from the stroma, that means the number of protons in the stroma decrease. Then obviously there will be an increase in the number of protons inside the lumen of the thylakoid. Till here, is it clear to you all, beta? Or these 3 points which increase the hydrogen ions inside or the protons inside the lumen as compared to the stroma. These 3 points are clear, or shall I repeat any of them? Yes, Priya is saying it is clear. Yes, beta. To anybody else? Yes, bache? Anybody who wants me to repeat any of the points? Okay, Yashika is also saying. Yes, bache? Yes, beta. I have got only 2 responses this time. Yashika, please take screenshot of the attendance and send it to me. Yes, beta. Deepika, Ananya, Ankita, Amrita. Okay. So these are the 3 methods by which hydrogen ion concentration or the number of protons they increase in the lumen of the thylakoid. Okay. Okay. Now then how this ATP will be synthesized? Now we have to come to one enzyme which is placed here again on the membrane. And that enzyme which is placed here on the membrane is ATP synthetase. In some books, it is written that ATP synthetase has just one part, F1, whereas F1 particle or F1, whereas in some books, it is written that the ATP synthetase has 2 parts, F0 and F1. So we go by NCERT where it is written just one part, which is F1. Now here, there are transmembrane channels. Here are transmembrane channels. Transmembrane channels means which allow the movement of ions through them. Channels, those are through the membranes. That means they are placed on the membrane. So they allow the movement of ions. They allow the movement of molecules through them. So this transmembrane channel which is present here is F0. F0 which is like this, or this is a type of protein. We can draw it little this way. Yes, this is F0. This is a type of protein. When we did fluid mosaic model, we did intrinsic protein, extrinsic protein, tunnel protein. This is same type of protein. So this is F0, which is a type of protein and forms the transmembrane channel. Transmembrane channel. So you must have come to understand what is this. This is a transmembrane channel, which is a type of protein or intrinsic protein. I'm writing here for you transmembrane channel. This is transmembrane channel F0. Then the enzyme which is required for the synthesis of ATP is known as ATP synthetase. See, the name is quite easy. ATP synthesis, synthetase. A is always used in enzyme. This suffix this suffix ase is always used to represent the names of enzymes. So ATP synthetase, that means the enzyme which helps in the synthesis of ATP. It has a part, it is here, and that is known as F1. This is F1. Or we can say ATP synthetase. You can also write down that ATP synthetase has 2 parts, F0 and F1. F0 forms the transmembrane channel. So this is ATP synthetase, which I have written 2 parts. Now what happens? Through this transmembrane channel, which is ATP, through this transmembrane channel, which is F0, through the transmembrane channel, which is F0, the protons which are more in number inside, these protons which are more in number inside, because